Hey darts fans, are we in Shanghai? No, we're on the Good Ship Undarted Waters. Hi, I'm Timo Newman, the transporter. Welcome aboard. It's a bit parky here with autumn coming on, so I'm just going to pop this jacket on here. Can't really throw in a jacket very well. Maybe there's a video there about the weight of clothes on our bodies and how that affects our throw. But anyway, not for today, because today is episode 3 of the series Philosophy and Strategy of Improving Performance. And in the first video we talked about optimization as a scientific way to think about improvement, and we kind of broke down a throw into what ended up being 11 different elements. And we talked about, conservatively, there were like 350,000 different throws when you looked at all the combinations of those 11 elements. I talked about that as an 11-dimensional space, which by coincidence is the space of string theory, the most sophisticated theory that physicists have ever written down to try and unify gravity and quantum theory. So we're in dizzy places here, you know, and this is pretty hardcore stuff. In the second video, I talked about a physics kind of way of thinking about optimization, and I used the analogy of annealing, which is how you make glass and steel ductile instead of brittle. And we saw the energy landscape, which is kind of the upside down version of what you might call the accuracy landscape in darts. Now, funnily enough, I was originally planning to have 2 videos on kind of the maths and the physics of this, and it ended up being one. And I was going to have one video on the biology of it, and it ended up being as many as you like, because when I started thinking about how to frame this in biological language, it really exploded in my head. And that's why I didn't make the video yesterday, because my mind was just buzzing with ideas, and I'm going to break it down into probably 2 videos. It could easily be 3 or 4. Depending on your appetite, really. But today and for the next video, we're going to talk about how can we think about optimization of throwing a dart in biological terms. It's not about the biomechanics of throwing a dart, it's about what can we learn from the natural world about optimization, because biology is all about optimization, survival of the fittest. That's the great maxim of Charles Darwin, the creator of the theory of evolution by natural selection, by far the biggest idea in modern biology, even though he wrote that down in the 1860s. So let me get my whiteboard. It's pre-prepared, and when you see it, it's going to be like, ah! And that is a very good reaction. If you don't go, there, there's something amiss. Maybe you're a biologist if you don't go, ah, when you see the whiteboard. Why should you make that sound when you see the whiteboard? Because there will be the language of genetics on that whiteboard. Now, I'm a physicist, but I worked on biology and medicine for many years. I had to learn basically biology in my 30s. And when I picked up a genetics book, I've never ever had the experience before of picking up a book that's supposed to be in English, and you read the first page and you literally don't know what any of the words mean. Biology is full of very precise terms. Physics is not like that. Physicists like to kind of talk very crudely, you know, in terms of pictures and whatnot, jargon. But the reason they can get away with that is in physics, in the end of the day, if you want to be precise, you write down the bloody equation. Excuse my language. You write down the bloomin' equation in physics. In biology, there are basically no equations. It's all about very precise language, classification. And in genetics, it turns out to be a very sophisticated subject, and so you have to have very precise terms. I'm going to use a very small number of those terms in order to make the connection, and it doesn't matter. So when you get this reaction, don't worry about it, but you'll follow along absolutely fine. But I needed to get it on the whiteboard first rather than waste your time writing it all. It takes a long time. So here we go. Here's the whiteboard. Let's turn out the old board light. Let's take out Shanghai, and here we go. And as I said, the natural reaction is, bah! But you know what? This is so cool. This is why we can actually make a lot of videos about this. So my aim today is to explain what's on the board behind me, and you'll really see the connection. And then we're going to relate that to what we talked about in the last two videos about this landscape. Okay, here's the egg box carton version of the landscape. All right, this is a two-dimensional landscape of peaks. In our dart throw, we've got an 11-dimensional space full of peaks and valleys. Okay, but this gives us an idea, and with the egg box, our tradition is that we throw it away when we've used it. Thank you very much. We're going to make a connection between this thing on the board behind me and that landscape, and you'll see precisely what I mean. And then we can talk about, well, in that landscape, which is called it's called a fitness landscape in biology. So we have a throw, we have an accuracy landscape in darts, we have an energy landscape in physics, and we have a fitness landscape in biology. We're going to relate this to the fitness landscape and see how does biology work in that fitness landscape. It's basically adaptation and evolution, and what can we learn from that and apply to our accuracy landscape, and it's going to be very instructive. Okay, so you've all heard of DNA and genetics and genes. You know, it's always in the newspapers, a gene for this, a gene for that. When we talk about genes in everyday language, we're really imprecise, and actually it's not the way that biologists talk about genes because it's too crude. Okay, so let me just be a little bit precise but relate it to things that everyone will be comfortable with. So the instructions that make an organism, and so the instructions that make us, so we start off as a fertilized egg in our mother's womb, and we grow into a multicellular organism of about 50 trillion cells. It's a miracle of nature, okay? But the blueprint for that is contained in the genes. Now, humans have roughly 20,000 genes, okay? And those are encoded in a molecule called DNA, and every cell in our body has the entire blueprint inside it, which is a remarkable thing in itself. So for humans, we have 20,000 genes, and they're on objects called chromosomes, okay? And we have 23 chromosomes. We actually have two lots of 23, but for our purposes today, our genome, which is the whole set of genes, is on 23 chromosomes. Now, what makes us different from each other? We all have the same genes, we all have the same chromosomes, so why are we all different? The reason is that for any given gene, there are many forms of that gene, which will be slightly different, and they'll give rise to slightly different proteins, and when those proteins operate in the cell, it makes us all slightly different, okay? Now, in the biology speak, when you talk about different forms of a gene, the word is allele, and that's why that's on the board behind me. You'll see the word allele. So let's make the connection now. So I've told you about kind of what we're all comfortable with, that we humans, we have 20,000 genes on 23 chromosomes, and we have, you know, some genes have maybe 2 or 3 alleles, so 2 or 3 variants. Others have hundreds or thousands of variants to do with the immune system, okay? So with our darts then, let's put all that terminology, let's put our darts throw into that. So here is the genome of a throw, okay? So this is the blueprint, not to make a human, but to make a darts throw. It has 4 chromosomes. It has a stance chromosome, an action chromosome, a grip chromosome, and a head chromosome. Now you could actually have more chromosomes because this is not unique, it's just a reasonable way to describe it. There are 11 genes, just like we talked about. We had a list of 11 things that made up the throw. In this biological language, that's 11 genes, and these genes have got different varieties. So for the first gene then is the orientation of our feet. So one allele of that will be that we're perpendicular to the line between us and the board. Another one is that we're an angle. Another allele is that we're head on. Those are different variants of the orientation gene, okay? And just like we counted up before how many different throws there were, in biology speak, you would say there are 350,000 ways of filling in this table. That they would call that genotype. So a genotype is an actual set of those alleles. So I've got a certain genotype. So you and I all share the same genome, but we have different genotypes. And the only two individuals that have the same genotype would be identical twins. Okay, so when someone says, oh, identical twins have the same genes, no, we all have the same genes, but identical twins have the same genotype. Namely, every different allele of every different gene, they share that particular variation, which makes them look and in some ways act very similar. Okay, we're more than our genes. That's a whole biology topic, because we develop in our mother's womb, we develop through life, our brain develops, our organs develop, and that's all perturbed by the environment. So we are much more than our genes, but our genes are the sort of fundamental blueprint. Right, so I'm not going to say any more about that, but I needed to get that across so we can sort of understand this table here. So what's on the stance chromosome? Well, there's the orientation of the feet gene, there's the feet separation gene, and there is the lean. How far do you lean forward or whatever? Are you upright or do you lean forward? Let me fill in. I'm going to fill in my genotype. All right. I've got actually a couple of different throws that I kind of favor, but the one that is best for me in terms of accuracy, I'm going to write down what my genotype is. So my feet orientation of the best one. So what would be the varieties, the alleles of that? It would be perpendicular to the line between us and the board, at an angle, or face on. Mine is perpendicular. Okay, so that's my allele for that gene. My feet separation gene, if I've got that particular orientation, I'm very far apart. So for this one, the alleles would be close together, somewhat apart, or very far apart. So my back foot is a long way back for balance, okay? And my lean is quite far forward. It's a medium lean. So low, medium, high lean. All right? Then we've got the action chromosome. That's got elbow height, wrist flick, the speed and angle of release of the dart, and that's got many alleles. I mean, in fact, there's many different speed and angle variations for which you can get an accurate dart, and that's going to be a whole other series about projectiles that we'll do shortly. And then you've got shoulder. It's only got two alleles in this simple model. Do you use the shoulder or not? So on my favorite action, then my elbow height is quite high. So let's call this low, medium, high. Wrist flick is very low, actually. I don't think I use much wrist in my darts, to be honest. I think I've got a pretty medium speed angle release. So if you had a high speed, low angle release, you'd have a very flat dart. If you had a lower speed, high angle release, you would have a very loopy dart. Okay, I'm definitely in the middle on that one. And no shoulder. I'm not into shoulder, okay? I'm anti-shoulder with my throws. We've got the grip chromosome. That only has two genes on it. It's got the number of fingers and the finger placement, okay? So I'm a very minimalist when it comes to grip. So I just have two fingers. So this would be 2, 3, 4, 5 fingers. I just have my thumb and forefinger. And then my finger placement would be, I would say, in the middle, you know, in the middle of the shaft. You can imagine this as front, middle, and back. I'm sort of in the middle there. And finally, the head chromosome just has two genes on it, which is the plane of throw. So is it lined up or is it lateral? Okay. I am on that particular stance. I would say it's not right lined up. It's actually, yeah, when you're doing that throw, you can't line it up. So it has to be somewhat to one side. So I'm just slightly to one side. And then the head tilt, probably low, actually. In other words, quite even. So for each gene, you have to actually say, well, what are the alleles? What are the different variations? So for example, for the wrist flick, it's like low, medium, high. For the number of fingers, it's like 2, 3, 4, or 5 fingers involved. Okay, so you you have to put a bit more information on this chart in a way about what are those alleles. It's like the alleles of eye color would be, you know, blue or brown or whatever, okay? But anyway, you get the picture. So that's my genotype. You know, you could say that's my DNA. That's the DNA of my throw, of my best throw. Okay? So let's get rid of this stuff underneath here. And I want to now use this to talk about how do we actually improve, all right? I want to relate this then to that accuracy landscape. So well, let's start with what we had right at the start. We said that our 11-dimensional space of all of those hills and valleys and ridges and stuff, we could project it onto just one dimension, and we call it accuracy in this way, and on here we just call it the 11-dimensional space. Okay, 11-dimensional space, and this would be full of hundreds and hundreds of peaks and valleys of all different kinds. And what we've talked about now a couple of times is that you want to be very high accuracy, but you might be stuck on a low peak. And that's what I talked about in the last video, that you might be stuck because you've started here, you've honed, but you can't get any better, because you're basically, whatever you change in a small way just keeps you on that hill. So when I talked about annealing in the physics analogy, what we talked about was turning this picture upside down to get to physics. So physics, we turn the board upside down, and this now becomes an energy landscape, energy going up that way, and nature likes low energy. So nature wants to be down here, but we might be stuck here in these metastable states, just like metal and glass, or steel and glass is when it's first formed, and then it's annealed. It's held at a higher temperature up here, so you increase its energy, you heat it up, you cool it down really slowly, and it then finds its way to a very nice low energy state, which in our speak, when we turn the board upside down, is a high accuracy state, okay? So how do we relate this to biology? Well, to get from physics to biology, we turn the board upside down. Right, so we turn the board upside down again to get back to biology. And in biology, you wouldn't call this accuracy, you would call this fitness, okay? You would call this fitness of an organism, and you wouldn't call this an 11-dimensional space. You would just basically call this the different genotypes, all right? So all the different varieties that you could fill in this this genome with its different alleles, okay? So we can now speak about biology very comfortably. An organism that has any chance of survival has to have a high fitness. Low fitness, you go extinct, okay? No organism has perfect fitness. There's always ways to improve. Okay, so that's a good lesson for us already, actually. So I want to just do what I did before with the physics picture and just make a slightly magnified version of this. So let's just zoom in to a few peaks. Okay, like that. And basically, as dart players, often we're stuck on these lower peaks, but we want to be at a much higher peak. That's how we make a really big step forward with our game. Now remember how I said that the beginner might start down here and they could hone their throw and actually go up to the top of the hill that they happen to be on. That's what happens to organisms too. So if you have a very simple organism like a bacteria, which has a very small genome compared to, say, humans, the way that bacteria can essentially get better is they have mutations, and you probably heard about mutations. So high radiation can cause mutations. sadly, we get cancer through mutations to our cells that can be caused by toxic agents, like cigarette smoke or asbestos and so on. Okay. Also, just actually most of the time by bad luck, unfortunately. That's a whole other story. But bacteria actually like mutations because that's the only way they can improve. All right. So bacteria then, when it divides into two new bacteria, there'll be some mistakes in the way it copies its DNA, and the new daughter cells will have some differences, those mutations. So what mutations do, it kind of allows you to jiggle around a bit in your genotype. It would be like on my throw that a mutation would be that this speed and angle of release gets a little bit shifted, or that my finger placement on the dart gets a little bit shifted. Those would be, in biology speak, point mutations. A single gene has been changed, and that will change my accuracy. Most of the time in bacteria, a mutation makes it slightly less fit. Those die, but now and again, a mutation makes it more fit, and then that fitter bacteria will have more daughter cells, and they will take over the colony. So colonies get better and better through chance mutations, and the mutations that favor certain individuals then spread through the colony because those individuals have more daughter cells because they're fitter. Fitter means that you leave more progeny behind. In darts, fitter means you win more games and you're invited to more tournaments. Okay, so this honing in dart speak, honing your throw, is kind of like a bacteria getting to the top of the kind of the fitness hill that it's on through mutations. It happens all the time. It's perfectly natural, but you know what the problem with that is? It doesn't have any innovation. It's like the most basic way that you can improve fitness. Okay, so what we want to think about in biological terms is, well, how does biology actually explore this space more? And one way that that can happen is to really change the genes. And bacteria even do this. Bacteria have a way of basically just picking up random DNA from their environment and inserting it into their own genome, which is a bit icky, really. They can also exchange DNA with each other in a process called conjugation. Okay, so there can be big accidents when DNA is copied, where large chunks are introduced or chunks are reversed. So biology explores this landscape by basically accidents. So in physics we talked about annealing, but in biology it's basically accidents for simple organisms where they just make really big errors, or they really just exchange lots of DNA with another cell. Okay, usually that's pretty harmful to them, but occasionally they get a big innovation. So just like with the annealing analogy last time, what we need to do in order to explore the space to find a better peak is to take risks. So these point mutations are kind of low risk but low reward, whereas if you change things up a lot, you have high risk because it's likely that the thing you've got now is not very good, but actually occasionally it's brilliant. So you have high risk but high reward. Okay, so one of these accidental things in DNA for my throw would be to do something like take these alleles in this action and just flip them round. So we basically just say, let's turn it upside down. Let's take this to here, this to here. That one stays the same, and maybe let's just take everything, just flip it all. That would be kind of like a major disruption to my DNA. So I could just do that. I could just take my genome, flip it around a bit, and actually say, right, now I've got to throw in this certain way. I've now got to throw with a low elbow height, with a lot of wrist flick, and more fingers. Now, of course, that's going to be horrible, and it will be basically, I'll have low accuracy. But as I was saying with the annealing, by exploring this space, even though it doesn't feel comfortable and it's low accuracy, we will discover things about our bodies that will help us. And when we revert back to what we thought was our old throw, it won't be our old throw anymore. elements of this will have been retained and we'll have a better throw, more accuracy, more resilience, and so on. Let me stop there. We're not done with the biology topic because there's two things I haven't really talked about in this, right? And they're both kind of important. So you could say, well, hang on, humans or mammals don't get a lot better by accidents. Accidents are bad because accidents in our DNA cause cancer and so on. And you're absolutely right. How do bigger organisms innovate? It's the S word, it's sex. Okay, let's be more poker faced, reproduction. So having a mating with another individual, whether that's in elephants or in humans or in snails or whatever you like, is a huge innovation in biology. It's thought to have kind of come about maybe a 500 million years ago, but it allows you in a much safer way. It's still pretty dangerous, by the way, out there in the wild. But anyway, because for all sorts of biological reasons, sex is a high-risk activity. You make yourself very vulnerable to predators and to disease, okay, out there. Snails don't have condoms. I'm sorry, but I mean, that's just how it is. In any case, it's relatively high risk for the organism, but much lower risk than just jumbling your DNA, but you have huge innovation. Okay, so I'm going to talk about, well, what would sex look like for our darts genome? Okay, obviously this can really go off the rails, so I'm going to try and keep it legal and good, okay, in the next video. But we're also going to talk about a really, really important thing, and this is actually crucial, is that evolution can only happen with challenge in an environment, and the environment in our throw is our darts. These are the environment. We have to interact with these, and these can interact with us. So those are going to be the two topics for the next video on this amazing topic of biology of the darts throw. So with that, crazy stuff. I wish you all well. I hope you're all doing well. Thank you everyone for your support. I wish you all the best. 3 darts at the Oki. Make them count, and I'll see you next time.